I wanted to quickly go over some aspects of meiosis and human reproduction to help us keep up to date so that we can stay on track with the exam schedule. So we last talked about chromosomes and aging and how there are a few hypotheses and theories to explain aging. I wanted you to be familiar with a few of them, namely that the ends of chromosomes have small segments called telomeres that shorten with age. With human reproduction, there are a few structures that we won't necessarily test you over in lecture, but that you need to be aware of. You should read through this chapter, chapter 26, or whichever version of the textbook you have, and kind of familiarize yourself with some structures and the function of structures. And so we talked a little bit about how with the SRY gene, how during embryonic development, the embryo develops external genitalia. And so that means penis, testes, prostate, and then there are associated ducts and glands. So I won't necessarily give you too much detail, but be aware of certain ducts, such as the seminal vesicle, prostate organ here. Notice how at the bladder, the urethra moves through the prostate, as well as the ejaculatory duct moving through the prostate, combining to form one duct in the urethra that exit at the end of the penis. The testis, where the maturation of sperm occurs, they have to hang out in the epididymis and then move up through the vas deferens, goes up and around to then join with some of these ducts before re-entering, or the contents entering into the urethra. And so just be generally aware of that, as well as the structure of the penis, the erectile tissues, etc. Read through that section of the book. Here it is in a cadaveric specimen. You can see this sagittal cut here. Again, just be aware of these types of structures for your future courses. Here's a different sort of coronal section here with the bladder, seminal vesicle duct. You can see here in these perineal areas, the different muscles associated with this, different glands, and then the nerve roots and erectile tissues. Here's a histological view of the inside of the testis, where you can see the seminiferous tubules, where meiosis is going to occur. The immature sperm will form, where the flagella develop from those microtubule structures, and then head to the epididymis. Again, just be aware of these structures, be familiar with them if you ever were to see them in your future, where they would be, what the tubules look like. Same thing with female organs of reproduction and surrounding structures. Please be aware of the uterus, lining of the uterus, the vaginal canal, the cervix, as well as the placement of the bladder below the uterus and the urethra exiting the bladder here. Understand the external anatomical structure, so the labia majora, minora, mons pubis, as well as the internal, so vaginal canal, vagina, bladder, uterus, rectum, etc. Be aware of these structures, especially where the ovary sits up above the uterus and these individual compartments. Again, here's a cadaveric specimen, sagittal cut, showing you rectum, ovary up here, probably much smaller than you think, uterus, bladder, urethra, vaginal canal. This image shows the uterine tubes. So notice that while connected, not necessarily, the uterine tubes aren't necessarily continuous with the ovary itself, but the oocyte has to release from the ovary and be swept up into the uterine tube to pass to the uterus itself. This smooth muscle-lined organ where the placenta and developing embryo will grow if fertilization and implantation occur. Be aware of the cervix, cervical canal, vaginal canal, etc. Here's histology of the ovary. We're going to discuss different types of follicles, as well as the egg structure, the oocyte that bursts from ovulation. We're going to discuss what happens to the follicle, how it becomes the corpus luteum, and the associated hormones. I won't go into too much of this. We talked about it in class a bit when we're looking at chromosomes. Please read through here and be familiar with the SRY gene located on the Y chromosome that basically has information right during embryonic development that tells the embryo to develop testis and stop developing ovaries. That SRY gene may be deleted And so an individual may be born XY but not have the SRY gene. So they would develop uterus, uterine tubes, etc. And the SRY gene may also translocate to an X chromosome. So individuals may be born XX but have the SRY gene. So externally, or so anatomically, they would have penis, testes, prostate, etc. Again, one of those things, it's not always as simple as XX, XY. It's more information here if you'd like to read. I basically test about what the SRY gene is and what it codes for. This is just kind of show you through cartoon images how during embryonic development, we have these undifferentiated organs, structures that could become the uterine tube or testis. Same thing externally, what we call genital tubercle and folds that can become the vagina, or during organogenesis with the SRY gene indicators become penis, testis, etc. Not necessarily need to know too much about this, just be aware of how that happens, and that things can also occur where individuals may be born with a combination of the two genitalia, or other such situations where the urethra itself doesn't fully close, or the tissue doesn't fully close around the urethra. And so individuals can be born with what's called hypospadias, which is urethric openings under the glans penis at the bottom, or on the top, epispadias. And that can also occur in various forms in the vagina, but it's less noticeable. It's usually fixed with surgery. Meiosis is a form of cell division. I need you to read through this section and understand meiosis. The only difference here is we are creating a cell, either a sperm or an egg, or an egg that could potentially combine, and we need that to get to 46 chromosomes. We talked about humans do not do too well with more or less than 46 chromosomes. And so we have to have a way to reduce the chromosome number. And so go from 46 to 23 chromosomes in the sperm. Same thing in the egg, go from 23 or from 46 to 23. And so meiosis is reproductive cell division. And so in the human, you're going to find this only in the egg and sperm cell, or what we can call oocyte and spermatocyte cell production, or oogenesis and spermatogenesis. So this is occurring in the testis and ovaries only. It's a reduction model. Meiosis, the older terms used to be reduction, chromosome reduction and division. And so we could say these only occur in the gonads, results in a cell with half the number of chromosomes, what we would call haploid as opposed to diploid. Here I'm just repeating myself, spermatogenesis and oogenesis. And so in our parent cell, we have 46 chromosomes, which is diploid. Each has a duplicate pair of chromosomes, one copy from one parent, one copy from the other. In meiosis, you are mixing those up and creating a cell with only one copy of each chromosome. So one of chromosome 1, one of chromosome 2, of 3, of 4, etc., all the way down to X and Y. These cells that we create, the sperm and the egg, are referred to as gametes. Gametes. So this whole process could be called gametogenesis, and they are haploid, 23 chromosomes. Spermatogenesis is occurring in the seminiferous tubules. You've already seen this picture. You called it testis, but really you're looking at a cross-section of a seminiferous tubule. And so here, so this is happening by the millions, right? So all of these sperm are created, and they will eventually kind of mature here in the epididymis before moving up into the vas deferens. Also notice in this chromatochord for later, during the organ version of lab, or the organ portion of lab, all of these veins, arteries, and nerves that run through here. The stem cell that can become a spermatocyte is called a spermatogonium. It is diploid, and it goes through mitosis. After going through mitosis, one cell remains, the spermatogonium, to divide again if needed. The other continues through this process of meiosis. And so the cell enlarges and moves toward the center, going through the process of meiosis I and meiosis II before eventually maturing into a sperm cell. And spermatogonium, or the gonial cell, is this sperm cell here at the base of the testis, or seminiferous tubule, I'm sorry. The primary spermatocyte divides. This primary spermatocyte is diploid. It rose from the spermatogonium. It moves toward the middle of the seminiferous tubule, what we call the lumen. At this process, it goes through a first round of meiosis, and rather than all of the chromosomes being in one line, the homologous pairs line up, what we call synapsis. And so this would be chromosome 1, chromosome 1, chromosome 2, chromosome 2. They line up next to their homologous pair, their homologous partner, a process called synapsis, different than mitosis, where they all lined up in one row. Meiosis, the homologous pairs line up, forming what we call a tetrad, 1, 2, 3, 4 versions of this chromosome here. Here's where you can get some genetic mixing. Crossing over can occur where the information from one chromosome crosses over onto the other. It's a process inside of our cells that mixes up our own individual DNA. It gives us genetic variability. Crossing over is where, here they show it in the red and yellow, where a portion of one chromosome mixes to the other. And so you're already mixing up genetic information inside of your sperm or egg cell before we even talk about fertilization. And so pieces of these chromatids can actually jump in a process called crossing over. It can happen once or none, but it increases genetic variation in sexually reproducing offspring. I'm going to pause for a second while the 12 o'clock Monday sirens go off. Probably hear that in the background. Okay, now that that's finished. Crossing over essentially is just where the information on one chromosome, on a homologous chromosome, will cross over to another homologous chromosome. This is not a mutation because it is essentially the same chromosome, just different, what we call alleles, a different version of that chromosome. And so it just increases genetic variation in meiosis, which could possibly be a test question, such as which process increases genetic variation during meiosis, A, B, C, or D. You will choose crossing over. This occurs when they line up during metaphase of meiosis 1. And so this first reduction, the first reducing split of this cell is meiosis 1, where this primary spermatocyte then divides into 2 secondary spermatocytes. There's no duplication of DNA because here our goal is to reduce the number of chromosomes. And so really our homologous pairs have been separated. And so if this is chromosome 1 and this is chromosome 3, as an example, this cell not only has one version of that, one version of that, one version, but because they are in that X shape, that's really 2 pairs of information. And so we're not done. We have to go through a second round of meiotic division. And so meiosis, in comparison, the first round of meiosis, meiosis 1, crossing over can happen. Your homologous pairs line up on the metaphase plate. Does not happen during mitosis, because during mitosis, you want the cell to have the exact number of chromosomes, not a reduced amount. But in meiosis, we're trying to get a reduced amount. And so we end up with these secondary cells, in this case, secondary spermatocytes, that still have too much chromosome information, but only one copy of that chromosome. And so the secondary spermatocyte is technically haploid, but we then have to go through another round to divide that chromosome to make sure we don't have too much genetic material. So we go through another process called meiosis II, where those secondary spermatocytes go through another round of division to separate into 2, or in this case 4, because both cells will be going through this, haploid cells with only one set of chromosome information. So meiosis 2 is the second round of meiosis, splits down the middle, splits those centromeres, and you get little spermatids. They're a circular cell. They are not a sperm yet. They have not matured, but they are a circular cell called a spermatid that can mature into a spermatozoa or into a sperm cell. The sperm itself, after maturation, is a cell that contains the information, contains the chromosome information in the nucleus, has mitochondria. This microtubule structure grows out into a whip-like flagellum. And so it has this head with an acrosome, this cap, that contains enzymes that could be used to penetrate the egg, and tons of mitochondria here to generate ATP so we can have movement of this flagella, this tubule structure. Inside of the spermatic tubules, we also have cells called sustentacular cells, or an easier word is nurse cell. These sustentacular cells are associated with these daughter cells, with these early spermatids, and they protect the sperm from the immune system so that our cell bodies don't recognize them as being foreign to us. Because with this maturation process, with this change, they do begin to trigger some immune symptoms of, hey, this isn't our cell type, this isn't our glycocalyx, something isn't right. And so these nurse cells kind of make sure that these cells can survive our immune system, or they don't trigger the immune system. But they mature in about a day. They can hang out from 1 day to 10 days before they're reabsorbed. They would mature, however, not inside the spermatocord itself, or I'm sorry, inside of the seminiferous tubule itself, but in the epididymis outside. So just kind of a general physiology behind this. The information to go through this process comes from the pituitary gland in the brain. So you have hormones such as the gonadotropic releasing hormone and luteinizing hormone that are released to trigger some of these cells to begin to produce testosterone and then begin the development of spermatocytes, spermatogenesis. This process starts at puberty and will continue for most of this adult's life. And so sperm production occurs in the seminiferous tubules. The spermatogonium goes through 1 round of meiosis, then 2 rounds of meiosis, and then this spermatid has to mature into a sperm cell. And the maturation occurs partially in the lumen of the seminiferous tubule and in the epididymis. Notice the sustentacular nurse cell right here in this image. The epididymis itself is above epididymis. Epididymis is above the testis itself. Has tons of little, they're kind of similar to cilia, but they are a different tubule called stereocilia, long branch microvilli that just help increase absorption so that you can kind of break down old sperm and other materials in there. Oogenesis is very similar. It's the production of the egg cell or the oocyte, and this occurs in the ovary, usually 1 ovary at a time. 1 egg cell will develop into a mature oocyte once per month, once every 28 to 40 days, usually just 1. However, most of this process, most of the development of the oogonium into primary oocytes occur in embryonic development, not puberty. This process starts in the embryo. The primary oocyte develops, most of the primary oocytes that will ever occur develop kind of along here in the ovary during embryonic development. And then during puberty, 1 of those, 1 or 2 are chosen per month to finish up the maturation process. And so it's kind of a halt in development. It's different than the production of spermatogonium. I'm sorry, the production of sperm in the spermatogonium, as in this occurs in embryonic development, whereas sperm production occurs after puberty. Same thing, however, though, you start out with a diploid cell, has to go through 1 round of meiosis to give you a haploid primary oocyte. During fetal development is where the process of meiosis 1 fully begins. The oogonium becomes a primary oocyte. After puberty, the primary oocytes complete a round of meiosis 1, but may never actually complete a 2nd round unless fertilization occurs. Another difference between oogenesis and spermatogenesis. So just kind of comparison here. The oogonium, the stem cell, goes through meiosis. This cell can go through 1 round of meiosis during embryonic fetal development, remains inactive until puberty, and then each month from puberty to menopause, 1 of those oocytes is kind of brought from that arrested, halted state and continues during that process of meiosis. However, most of those cells are formed during embryonic development. They begin the process during fetal development, halt, begin again 1 at a time during puberty. The control mechanisms are the same in ovary development as opposed to, or not opposed to, is the same as spermatogenesis. Gonadotropin releasing hormone, luteinizing hormone is released. We'll get a little bit more detail on these hormones after we're done with meiosis here. But just know that the pituitary controls the process of this development of oogenesis, causes several follicles to grow, etc. I'll go into this more detail in class. The development of the follicles themselves. The follicle is what is around the ovary or the oocyte inside of the ovary. And so after puberty, when those hormones are released, one follicle is chosen, 1 to 2. The primary follicle begins to grow, and that oocyte grows inside of it as it goes through a round of meiosis. Or finishes up meiosis 1, I should say. The secondary oocyte is what forms if the cell goes through this process. And so what you'd find in the secondary oocyte is a large follicle with an egg cell and a ton of cytoplasm. During this round of meiosis, when you divide the chromosomes in half first, you get one secondary oocyte and one polar body. A polar body is just a small structure that acts as a place to put the chromosomes. It's often absorbed, dissolved. It sometimes can go through another round of meiosis, but rarely does it do so. It's just sort of, you're putting all of your energy into one egg cell. In spermatogenesis, you put everything, you made 4 sperm cells. In oogenesis, the goal is to make one large egg cell. And so here it is in picture form. Just like in spermatogenesis, you begin with the primary oocyte. Most of those, I said, were formed and began the process of meiosis in embryonic fetal development. When you go through the round of meiosis 1, you end up with one large oocyte and one secondary one polar body that's often absorbed. It's just a place for those chromosomes. Now, if it goes through another round of meiosis 2, so if this cell is fertilized and it goes through meiosis 2, then yes, you would get 3 other structures. But unlike in spermatogenesis, where you got 4 sperm cells, you're creating one large oocyte, what we call the zygote if it's fertilized, and a couple, 4 in this case, polar bodies that are only a location for those chromosomes. They do not form an egg cell. They do not fertilize. They're often absorbed or dissolved. Now, if fertilization occurs, make sure to highlight that, underline it, look at that. If fertilization occurs, then the oocyte will go through the second round of meiosis. If not, it never does. It's just released during menstruation. But look at this picture to help you. You have the oogonium, primary oocyte. After puberty, the primary oocyte completes meiosis 1, because remember, it was halted during fetal development. So you get a secondary oocyte and a polar body that's probably going to be dissolved. Now, if the secondary oocyte, if it is fertilized, then it will quickly go through a round of meiosis to separate those chromosomes so that you'll only have one set fused with the sperm. Those other chromosomes are put into a secondary polar body that is dissolved or absorbed, and then you end up with a fertilized zygote. So if fertilization occurs, at the end, you would have one fertilized zygote and 1 to 3 polar bodies. Again, I'm just repeating myself here, but polar bodies are just a location for that chromosome during division. You only end up with another round if this oocyte is fertilized. This polar body may also go through meiosis, but you're not forming another oocyte. You're just forming a small polar body. Technically, because the fertilization has occurred at the end of meiosis 2, you call this an ovum, a fertilized egg cell, ovum, and these chromosomes from the sperm will fuse with the chromosomes from the egg, forming a zygote, one cell, the beginning of a human. The end result of oogenesis is a diploid ovum because the sperm brought its chromosomes with 3 polar bodies. This is a test question. The ovum is fertilized to become what we call the zygote. When those chromosomes and the chromosomes from the ovum fuse together, it is now a zygote. And so take a look at sperm versus oocyte formations, spermatogenesis, oogenesis, understand the difference, how you always end up with 4 sperm at the end. However, in the egg, only if it's fertilized do you get 1 ovum and then 1 to 3 polar bodies. That is about the round I would like you to understand. We'll finish up the rest in class. Thank you and happy studying.